Hello friends of plus 2, continuing the first lesson of the second unit, the genetics and evolution. This unit has 3 lessons: principles of inheritance, molecular basis of inheritance, and evolution. The first lesson is at its climax, and yesterday's topic I gave you to study about mutations, types of mutation, numerical changes in chromosomes, and today's topic is methods to study human genetics. What are the methods how we study? Mendel gave us many guidelines and steps to study genetics. And the one wonderful thing about the Mendel was that during his lifetime, nobody recognized him. This is the tragedy of India. Indian culture is such a beautiful culture that nobody likes when a person is alive. When he dies, all people like him. When he was living, nobody recognized him, nobody knew that who was Mendel and who. Mendel published their every result in the newspapers. And after the death, 16 years of his death, the other scientists recognized Mendel and named the Mendel. Today we know the Mendel. After his death, he does not know that his rules and regulations and principles of inheritance are applicable to human beings. No. Topic is how to study human genetics. Mendel gave the guidelines. We can study the human genetics by pedigree record. Number one, by the studying pedigree analysis of a particular disease of the forefathers. Number second, study of population genetics, family planning methods. Third, biochemical genetics, that is the chemical basis of life, that is the DNA and RNA. Cell culture technique, somatic cell fusion techniques, that is study of characters of the twins, that is genetic behavior. Then phenyl thiocarbamine is a chemical. Our differences of individual differences due to difference of chromosomes and genes. Some are tastiers of chemical PTC, phenyl thiocarbamine. Some do not taste. That is a tasteless, a bitter chemical but harmless. Albinism, absence of melanin genes. Some people are white in color, no black pigment due to absence of that gene. Then color blindness, albinism, that is called rolling of tongue like this. Can you try it? Some persons can roll their tongue, they are rollers. Some person they do like this. They cannot roll their tongue. You try it. You can or cannot. That is due to genetics, genes. Then Bohr body is due to X chromosome in cell. We will explain. That is bending patterns of the chromosome by colorful dyes. They give the difference of chromosomes. That is we can study the chromosome and genes by the study of cells. Then mental retardedness and abortion, malnutrition, nutrition we can study by bending patterns of the child before birth, and we can recover the disease. The process I told you, amnicentosis by we can study the diseases of the organism. This is all about methods to study human genetics. Next part of the lesson is genetic disorders. Genetic disorders are of two types: Mendelian disorders and chromosomal disorders. Mendelian disorders are very simple: hemophilia, color blindness, red-green color blindness, you can say sickle cell anemia. These three are Mendelian disorders. And fourth is phenylketonuria, then cystic fibrosis. I will study all about these characters. Hemophilia. Hemophilia is symptoms are that the person who has no gene for coagulation of blood during injury. When there is injury, there is a plug or coagulation of blood. So then, if that gene is absent, no coagulation of blood, and the person dies when he has a cut or accident, no clotting of blood. That is due to gene. That is that is due to gene. That is a recessive gene present on X chromosome. Recessive gene present on X chromosome. If it is present, the person is hemophilic. Second, the male are more hemophilic than female because sex-linked diseases, I told you, in male there is only one X chromosome. It is present on X chromosome, that male is hemophilic. In female, if it is present on one X chromosome, female is carrier. Male is never carrier. If it is present on two chromosomes of the X of the female, then the female is hemophilic. So hemophilic person dies at the earlier stage. These diseases 5 I will discuss in tomorrow lesson. The hemophilia I told you yesterday, I will revise. Red-green color blindness and sickle cell anemia, phenylketonuria and cystic fibrosis I will explain in tomorrow's lesson. You study every topic very carefully. Thank you.